I've just put a picture up here to orientate you. The University of Queensland is based in Brisbane on the southeast coast. So we're just across the other side of the Pacific. So as Dan mentioned, I come from Naomi Ray, Peter Vischer and Jian Yang's group. So the group is focused on complex traits. I came to the group with a background in more molecular genetics. So I did an undergraduate actually in sports science, and then my PhD was in a neuromuscular lab, and so we were just modelling a normal variant in a knockout mouse. And then we did some overexpression studies. So I kind of come from a wet lab background. At the moment I'm looking at motor neuron disease, and that's a complex neurodegenerative disorder. So I guess that's a bit of background about what I've done and so where I am at the moment. And so a lot of these foundations I've kind of picked up along the way, and I guess that's because the field is constantly changing, particularly with new technologies, as you heard about in Dan's opening lecture. So with that today I'm going to be talking about real basics. So what is a cell? What is a gene? We'll talk about transcription and translation, and then talking about cell division and Mendelian inheritance patterns. So I've put this together with mainly this source material and also a bit of help from the guys in my group listed there at the bottom. We had an economist, Ronald, who came and spent some time with us. So he actually gave some advice on what would be good to know, or what he wished that he'd heard about before he kind of was launched into the genetics field. So I'll just start with the cell theory of life. So in 1665, Robert Hooke was looking under a microscope and he defined the term cell. So this comes from the word cella, which just means it's Latin for a small room or with walls. And then in 1838 and 1839, Schleiden and Schwann defined the two facts that, or theories, that all living creatures are made up of cells, and the cell is the smallest unit of life. And then it's a bit controversial on who came up with the third. All cells arise from cells, but I think Robert Remak is the earliest that can be determined. So there are two main types of cells. So I guess we tend to think of things either as prokaryotic or eukaryotic. An example of prokaryotic cell is bacteria. So bacteria has single circular strands of genetic material that floats free inside the cell wall. Typically these are small unicellular organisms. They live in colonies. They reproduce asexually, as you heard in James' talk. And interestingly, they can also be found in extreme environments. So they also live on multicellular organisms. And so multicellular organisms, we're typically talking about eukaryotic cells, so things like yeast, plants, and of course humans. So these cells are highly organized. They're a lot more complex. They have a lot more structure inside them. The genetic material is housed inside a nucleus, and then of course, given that they're a multicellular organism, this gives them the opportunity to specialize. Which we'll talk more about in the later lectures tomorrow. So DNA, or as you know from the trivia, deoxyribonucleic acid. It was defined in 1944 using a bacterial gene transfer that DNA is actually what carries the genetic information. Oh.
It's 5642.
Okay, hopefully that doesn't keep happening. Okay, so, and then as we also know, Rosalind Franklin showed Frances Crick and James Watson her X-ray crystallography picture, and they were able to deduce that DNA is actually in the form of a double helix. So as you can see here, or a spiral staircase, where you've got matched pairs of bases that run opposite to each other, so it's in an anti-parallel structure. Here's a more descriptive picture of the DNA. So you've got a phosphate deoxyribose backbone. Here are the bases A and its complementary base T. And these are connected by hydrogen bonds. So the DNA sequence is essentially just the order of these nucleotide bases. So all being made up of adenine, thymine, cytosine, and guanine. Okay, so in the cell, this is just an overview of what information gets processed and where. This is the DNA, so it's in the form of what you can see as a compact chromatin. DNA gets transcribed inside the nucleus, so this is where an RNA polymerase uses DNA as a template to synthesize RNA. Then this RNA actually gets exported outside of the nucleus, and this is where translation happens. Okay, hopefully this doesn't happen again. All right, so I was up to, so basically the RNA gets transported out, and messenger RNA gets translated into proteins. So, I mean, when you're analyzing the DNA, we generally communicate in terms of genes. So what is a gene and how is it defined? So basically a gene is just part of a DNA, which is a template to make RNA. So this can go on to be messenger RNA or things like non-coding RNA. When we talk about genes, these can either be RNA genes or protein coding genes. So RNA genes get transcribed only. So, and there's about 10,000 of those that have been annotated so far. And then we have protein coding genes. So there are about 21,000 of those. And these get transcribed and translated into the protein.
What does that mean? What's the difference between those two things? Transcription and translation?
So there's jobs for RNA that don't require it to be translated into a protein. So things like transfer RNAs, ribosomal RNAs, microRNAs, small nuclear RNAs, all are transcribed from the DNA, but don't get translated into a protein. Okay. Into amino acids, they don't get, they don't become a long peptide amino acid chain. So they just stay in their RNA, and they have a function in that form.
Okay.
Okay. So in total, there's about 30,000 genes, and only 21,000 of those are protein coding. So this is a typical DNA annotation of what a gene looks like. So these are, consensus type regions where we'll have a promoter region, so that tells, that is a signal for transcription to start. Then we'll have a leader region before the start codon. So here's shown by an ATG. Then you'll have typically an exonic region, an intronic region here in grey, followed by however many more exons and introns. And then this is, there's a stop codon to indicate the end of the transcript, a end sequence, and a site that tells the mRNA to be cleaved. So this is the general format. Of course, it's not that simple to just define a gene based on an in silico analysis. Yes?
Okay, so what's the difference between the promoter and the start codon, and what's the difference between the end codon and the stop transcribing?
So the promoter is just a signal that can vary between, that tells a transcription machinery to come bind here. And the start codon is the start of the actual, let's say it gets translated into a protein, it's the very start of that first amino acid. Yet that promoter region actually would be cleaved off and got rid of. It doesn't play a role in this, for example, the protein coding, the final result. So.
Can you say that again but point to the picture from before?
Yes. Okay, so this is the start of the exonic region here at this start codon. So this is where you start to read, once it becomes messenger RNA, this is when you start to read the bases in codons, and you have an amino acid that's added to each codon, but this region here is not actually part of the protein. But it's a region where you might have enhancers or silencers. It's like a transcriptional control region that can tell, that can provide like a form of regulation of gene expression.
Like the metadata, sort of?
Yeah, I guess you could describe it like that. Yeah, I mean, yeah, you can describe it like that. That's right.
And what is
So-
the intron?
So the intron typically in a protein coding gene gets cleaved out of the final protein. So
Is there anything that lets you know that it's going to be an intron, or we just know because we look at the proteins?
there is consensus sequences that tell you where the intron-exon junction is, and I'll talk about that as well.
Is the intron, does it have like promoter regulator functions like the promoter region, or what's the function of the intron if it doesn't get coded into the protein? Is it
That's a difficult question. I don't know. I can't, yeah, it's hard to say what does one particular intron do. We know that alternative splicing exists, and this is a very important way that the genome can actually, it's a very important source of diversity. So you might have alternative start codons, and regions might be skipped, or intronic regions. Yeah, I mean, I don't know. It's hard to say.
Can I take a stab at it?
Yes,
They were
please.
thought to be useless because people just assumed they were useless, but certainly some of them contain enhancers or microRNAs. At a minimum, they need to have this splicing code instructions so that they get spliced out properly, or you don't get the right gene.
Yeah, that's right.
weren't they before called junk DNA?
So there's no such thing as junk DNA.
It used to be called junk DNA.
Yeah.
Something different.
So could the intron be thought of as a type of promoter then, or what makes something? I mean, besides being in the middle, is there anything that makes an intron different from a promoter?
I'm sure there's things that make it different. For example, the consensus site that indicates for a spliceosome to chop it out. So that would make it different. But in general, I guess what's known about the metadata of transcription is that it occurs in a symmetrical region. So both on either sides of the start codon. So that's also interesting to note. But yeah, I mean, I guess we're in the very initial stages of trying to work out exactly what all of these regions are doing. And I'll bring up ENCODE, and so that's the Encyclopedia of Genetic Elements. And you know, there's a lot of groups that are looking into these non-coding regions and what role are they actually playing. Because they obviously are conserved throughout evolution, and so they must play a role somewhere. Yes, James?
Well, let me take a stab at it as well. So the promoter, which is immediately adjacent to the transcription start site, is definitely functional for all genes, and that's where RNA polymerase II, which is this, basically just transcribes the DNA into RNA, is going to assemble. In principle, an intron could do nothing. It could just be eventually excised once the messenger RNA is ready to serve as a template for translation. But as was said, in practice, it turns out that introns often contain regulatory sequence. I mean, the example that always comes to mind, the mutation that causes blue eyes, as opposed to the ancestral state, which is dark eyes, That mutation is actually located in an intron of a gene that is to the left of the gene that actually product is actually that actually encodes the pigment. So that's OCA2. But the causal SNP for blue eyes is actually located in an upstream gene called PERK2. I believe it's the last intron.
Yeah, there are examples of traits and diseases that are associated with these intronic regions. So they are playing an important role from that aspect. In terms of annotation, it's not just done in silico based on sequences, but it's kind of from both an automated and manual approach. So the initial gene map is very much experimentally refined through things like real-time PCR or RNA-seq to really confirm the existence of particular RNA sequences of the hypothesized genes. And as an example, GeneCode has developed an automated AnnoTrack system that contains data from, so these are a lot of different groups that are contributing to this resource, and this they've highlighted differences in the pipeline to kind of coordinate quality control and track outcomes. So this is the genomic sequence here in grey. As an example, RNA-seq here in orange. Proteins are here in green. The transcripts here in blue. And so basically these come down together to, inform what is a gene and what isn't. So it's not necessarily a straightforward process, and I think that's why it's constantly being updated and refined. And so particularly with these new next generation transcriptomics, hopefully with the drop in price, improve in error rate of longer reads, this will really improve the analysis of the transcriptome annotation and really help understand transcript structure within a cell and during its development. So how does transcription work? So as we've mentioned, DNA is made up of two strands, a template strand and a complementary strand. An RNA polymerase is responsible for pulling these apart, and then the RNA is able to provide complementary bases to create a long strand made up of exons and introns. And then the introns are spliced out to form messenger RNA. So I've actually got a movie to actually show you a more realistic picture of transcription. So this is in a free iBook actually, which you can download, and I really, the animations are actually based on as close as possible to being real. So that's why I quite like them. I don't know what's happening. Oh, thank you. Okay, so in purple, we've got DNA here in its double helix, and coming out here is the RNA. So, and this large thing here is the RNA polymerase, and so inside it's splitting up the two strands of the DNA, and you can see that the RNA is being added at each base. So this happens very, very quickly, and it's a very efficient process. And then it'll come across a consensus region where it will say, okay, stop here, and it will cleave off the RNA.
What was the name of that?
It's E.O. Wilson's iBook, and it's Unit 3. I'll put the slides on Dropbox, and then you'll have the link there for the source material. So I guess you've seen a picture of how the transcription is working. What I was trying to say is at the 3' end there's a consensus sequence, and this actually cleaves the nucleotides and cuts the RNA. A cap and a tail are pretty much added to this RNA, and then that kind of indicates that, okay, I'm ready to be spliced, and that happens outside of the nucleus. So some principles about transcription. As mentioned, there's no such thing as junk DNA. Majority of bases are associated with at least one primary transcript. Chromatin accessibility. So this is DNA is, you don't see DNA in a double helix. It actually is tightly compacted, because if you put all your chromosomes together in, against a ruler, it's about 2 meters in length. So it's very, very highly folded. But if it's getting transcribed, then obviously it needs to open up. So this chromatin, and it's called chromatin when it's all folded up. So this accessibility, and also histone modification. So this is like a methylation approach to changing the DNA structure without changing the DNA bases. These are highly predictive of both the presence and activity of these transcriptional start sites. And so this DNA replication is closely correlated with chromatin structure. And there's quite a lot of control mechanisms. So an RNA polymerase can't just initiate transcription anywhere along the DNA. They require regulatory factors such as a promoter. Promoters are recognized and bound by transcription factor machinery, really, that guide and activate this RNA polymerase. When we talk about transcription factors, so we're saying they're acting in trans because they're produced by a remote area of the DNA, and then they mitigate to a transcriptional start site. So that's their site of action. Promoters, we tend to say that they're cis-acting because they're located close to the transcriptional start site. And then there's also things like enhancers, silencers, and these are also cis-acting, so short sequence elements that can alter the transcriptional activity of a gene. Yes?
So, just unclear, from a couple slides back, it sounded like if you transcribe a gene or you're trying to map it out, basically what you're doing is you're trying to figure out all the pieces in the DNA that are functionally linked in some way, and that's...
Yeah, I guess that's why we talk in terms of genes, because that's how the DNA is partitioned to become a functional element.
But it can't, but like physically those pieces are in different places in the strand, potentially, right?
In terms of...
In terms of, the actual pieces of the DNA, like the actual base pairs. So a single gene can have base pairs that are in different places. They're not all following each other in the DNA sequence, right? That's what you're saying.
I think a DNA is generally considered to be a segment of the DNA that is all in one place.
Okay.
Maybe with longer read technology, we'll find genes that are, split apart, spliced together. But at the moment, as far as I'm aware, genes are just essentially in one area. But there are factors that can act on those genes to initiate transcription.
And those don't count as part of the gene
Yeah.
themselves?
Yeah, I mean...
The definition of a gene is, theoretical and, it's controversial. You're just different depending on your context, right? So,
Yeah.
Can I?
Yes.
So just related to what he was asking. So when you showed the picture, and you said here is the an intron, but it was it looked like it was in the middle of the gene, and so the parts of the gene that were actually coding the protein were separated. But would you still consider
Is this picture you mean?
Well, I was thinking about the one where you had all the letters up, but
Yeah.
So you have this intron region, right? But then you have the exon, but then those are going to be spliced together to be one protein when we're done.
That's right.
When you think about what a gene is, are you picturing from the start codon to
Yes.
the end codon, even though it includes the intronic region?
Yeah, yeah.
not-
I mean, that's still a gene.
Okay.
The
Even though
intronic
that's not
region
coding for
is still a gene.
Okay.
Yeah. Yes.
And here you only show one intron, but in the other
This is just
picture,
an example.
there were many more, no? In
Yeah,
the other
that's-
picture, okay, so there can be many of them.
Yeah.
Okay.
Yeah. And there can be different isoforms. So even a gene can have, in one particular tissue, it might encode 6 exons, and then in different tissue, the same gene might only encode 3 exons. And so that's a way of diversifying function from a single gene. Yeah. Is there another question? Okay. So we'll go on to translation. I'll talk about the spliceosome. So this is talking about splicing out. How do we know where the intron starts and the exon finishes? So we're aware of something called the spliceosome. So this is a machinery that consists of small nuclear RNAs and proteins, which can recognize introns by their splice site sequences located at the intron-exon junction. So this is a picture of an exon, the intron, exon here. And so there's 2 different mechanisms, U2 and a U12. U2 mechanism always has this GT dinucleotide and always tends to end with this AG. And when you see these types of pictures where you've got all these bases here, it's just an indicator. The size of the letter actually indicates the frequency that the base is seen. So it can vary, it could be a C, an A, a G, or a T at this particular one, but it's always a GT here. So this is an indication of the consensus site and how it varies across different genes.
What does consensus mean?
A common site. So
It's a consensus across genes or across what?
across genes, across intron-exon junctions,
So that
across
means
all
that-
things that have been measured.
So across
In
many
humans.
different genes,
Yeah.
this is now, there's a common signal that there are common
Yeah,
way to recognize.
And
the-
across
maybe it's a
many different
probability
organisms.
or
Yeah,
um,
like evolutionarily distinct in most cases.
So if you're interested in understanding how the spliceosome works, there's a video in the iBook. But essentially what happens is there's 2 binding areas. So this being the exon, and intron here. And then these 2 regions come together with a cleavage factor, and pretty much brings the 2 exons together, and the intron becomes a loop that is released into the into the cell. So I've mentioned this quite a few times. I guess this is more a picture for you to view that one gene can actually become multiple transcripts. So typically you'll have, for example, 3 exons and then the introns are cleaved out. But of course you might get alternative splicing, which means you might get a transcript that has these 3 exons, but you also might get a transcript that only has the first and last exon. You can have alternative start sites of the gene, alternative ending sites. So this is a major source of diversity, and this is why cells can actually specialize. So in terms of translation, I'll show you a video of this, but basically this is when you're going from messenger RNA to a protein. So 2 subunits of a ribosome bind to the messenger RNA, position the start codon to interact with that transfer RNA molecule. Amino acids are delivered to the ribosome by the transfer RNA. And the messenger RNA is read in codons. So this is three bases that make up each amino acid. And then the ribosome releases the completed protein chain as well as the messenger RNA, and the ribosome falls apart into its 2 subunits. Okay, so here is the ribosome, and you can see what's happening here is that the transfer RNAs are adding amino acids. So here's the messenger RNA. Here are the codons, 3 bases here, and each 3 bases indicate the amino acid that gets added to the messenger RNA by this transfer RNA.
And so earlier when you were talking about the difference between, there was like 30,000 genes, but there was 10,000 and 20,000. So 10,000 of them just stay like this, is what you're saying? Like they
Yeah,
stay
10,000 of them don't get translated into a protein. Yeah, and so the completed protein basically was the blue chain, and that chain actually then folded up into a kind of more like a secondary 3D structure. So this is what your amino acids actually are. So as mentioned, 3 base pairs makes a codon, which indicates which amino acid for the transfer RNAs to add to the messenger RNA. So there's 4 possible bases, G, U, A, C. This implies there's actually 64 different combinations of bases of codons, but there's actually redundancy in the genome in that there's only 20 amino acids. So these are the 20 here, and so each amino acid can be encoded by multiple different sets of codons. So for example, arginine has 6 different codon combinations, and so how you would read this circle is G, C, C. Well, that's an alanine. So why is this important? Well, there's there's consequences basically for different coding changes. So if the base change results from an arginine to an arginine, you're not going to expect a large functional effect. But if the amino acid has a change in polarity or charge, then it might have a more significant effect. Or maybe what you've gone from is a normal codon to a stop codon. So then that's a loss of function change, and the messenger RNA will actually stop being translated into the protein. So you can kind of make inferences about what the functional effect could be based on these coding changes. So I mentioned the amino acid chain actually has a structure. So there's alpha helix, beta sheets, beta turns, different protein domains that can make up this 3D structure. So this is an example of a secondary structure. It's folded up into a tertiary structure. And then of course you can get proteins interacting with each other to form, for example, a receptor. And so that's more a quaternary structure. So the majority of Mendelian phenotypes, so we'll talk about Mendel's inheritance in a few slides, but basically these are current. So this is one gene or one loci associated with a particular disease. The majority of these types of phenotypes are currently associated with protein coding changes. So these the protein coding region of the genome is only about 3 to 4%, but it's very much enriched for these significant variants that impact on function. So zooming back out of the DNA and just looking at more a macro level of DNA organization. DNA is organized into chromosomes. So chromosomes, I've tried to explain, but DNA essentially has a dynamic structure. It's as do chromosomes. They're actually situated in territories. So if this is a nucleus that's been fluorescently labeled, for the different chromosomes. And this seems to be evolutionary conserved, which is quite interesting. And so when they're carrying out transcription, so this is creating the RNA, creating the protein, they're actually relatively decondensed. So the DNA is more open. But when at cell division, so when the cell is going into, for example, mitosis or meiosis, the chromosomes are really condensed. So, and this is an example of it being condensed into these two arms. So human cells have 23 chromosomes. So this is a picture of it organized in terms of size. And so 22 chromosomes with 2 sex chromosomes. And these can be either diploid or haploid. Okay, so this isn't the end, but just a quick summary of what we've talked about. DNA is located in every single cell type. Most of the DNA is transcribed. Transcription factors, enhancers, promoters are all involved in regulating gene transcription. Alternatively, splicing of messenger RNA is a major source of diversity. Most of the DNA is regulatory or structural, so only 3 to 4% is protein coding. Proteins are made up of chains of amino acids, which form a dynamic 3D structure. Chromatin structure is related to gene transcription, and genes are organized into chromosomes, which condense to carry out cell division. So now I'll talk about meiosis, mitosis, and also Mendelian patterns of inheritance. So the main function of cell division is really for growth and repair. Cells have to remain efficient, and it's typically easier to divide rather than increase in size. Mitosis is an asexual cell division, which results in an equal number of chromosomes and happens in a fairly quick manner. Meiosis can take a lot longer. It's sexual cell reproduction and results in half the number of chromosomes. The advantage of meiosis, as James mentioned in his lecture, was that there's novel combinations of genes which are possible in each generation. Usually chromosomes are uncoiled intermixed, and small parts of the DNA are actually wrapped up in these histone proteins. So this is a nucleosome here. So the DNA is wrapped about 1.7 times around each nucleosome, each histone. And then during cell division, DNA is condensed and coiled into intact chromosomes. So this is just a picture of the different stages of mitosis. So we have prophase, metaphase, anaphase, telophase, and cytokinesis when the cell splits apart. I guess this is an organized process, and the chromosomes do align and pull apart during this growth and repair stage. I'll probably focus more on meiosis just because it's interesting in terms of it's a source of diversity. Meiosis is really a single round of cell replication followed by 2 rounds of duplication. What's important in this process is that you need homologous pairs for this process of recombination, and the resulting gametes are genetically unique. So germ cells proliferate by mitosis until they receive the right signals to enter meiosis. And so this actually differs between males and females. So females are born with a finite number of oocytes, and these are arrested in prophase 1. Male sperm, however, they don't enter meiosis until after puberty, and then that's when they actively divide. So just an example of crossing over. What do I mean by this? What actually happens? So in terms of a practical example, basically if there were 2 genes, one for eye colour, one for body colour, both on the same allele. So this actually does exist in flies. So here I've just drawn an allele in the terms of one line, and these are the two different genes. So we've got white eyes with a grey body, and then we've got red eyes with a yellow body. So the red eyes and the yellow body here in this situation are acting dominantly. This is on the X chromosome, so here's the male and this is the female. So the female has 2 copies of the X, the male just has 1 copy of the X. If you inherit one allele from each parent, basically you would expect, and you can actually draw a Punnett square to demonstrate this to yourself, but basically you'd expect about 50% to have white eyes and a grey body, and 50% to have red eyes and a yellow body. However, what was actually demonstrated in these experiments was that there was a proportion of flies that actually had white eyes and a yellow body and red eyes and a grey body. So this wasn't Mendelian segregation. And so what had actually happened here is that segments of the X chromosome had swapped places. So this crossover point alters the location of 2 genes. And so this would have happened in the female. So during meiosis, there would have been, the X chromosome would have lined up together. And then during this process of recombination, 2 areas of the genome would be broken by enzymes. Then during the repair process, they'd swap positions. And then this is when you'd get this scenario. So the crossover point, as I mentioned, can alter the location of 2 genes, and they can end up on different chromosomes. And so this generates an entirely new phenotype in the next generation of flies. So 2 points to make about recombination is that genes that are further apart are more likely to become separated during the crossover process, and alleles that are located close together are said to be linked. And so this is a diagram of the crossover happening. Here you can see this is the break between the 2 lining up of the homologous chromosomes, and here they've actually swapped positions. So it can occur anywhere along the chromosome, but in humans, in actual fact, it's quite common. So there's 1 or 2 crossovers or more, which is usual along each pair of homologous chromosomes. So multiple segments of homologous chromosomes can swap places. And so obviously the same genes are present, you're still inheriting the same, but they're just in a different, the combination of the genes changes.
Fleur?
Yes.
So all these things you're talking about, how common are they in non-human species? Like, is meiosis the same in like any multi-cell?
I'd say yes.
Oh, okay.
I mean, different organisms have different numbers of chromosomes, different sizes of chromosomes, and that would obviously impact how the probability of their chromosomes recombining. But as far as I'm aware, I don't see why it would be different. And this is part of diversity and evolution and natural selection. It's this that comes with sexual reproduction. Yeah. So for the last part of the lecture, I'll just talk about Mendel's laws. So Mendel was an Austrian monk who did a lot of pea plant experiments in 1866. It was a huge amount of work that he did, and it wasn't really rediscovered until 40 years later in the 1900s. So the way he did this was he used true breeding. So this is what's referred to as homozygotes pea plants for 7 different traits. So the offspring of this first cross would be heterozygous and would therefore display the dominant state uniformly. And again, you can draw up the Punnett square if you're interested. He used a capital and a lowercase letter to indicate the hybrid genotype, to indicate 2 alleles of the one gene. So the 2 main laws he concluded from these experiments was the law of segregation. So each parent has 2 alleles. These parent alleles separate and only pass on 1 copy of their allele to the next generation P. And in the law of independent assortment, which states that each P inherits 2 alleles that can determine its shape, or another trait, one from each parent. These do not blend. Either they have a visible effect, which means it's dominant, or they remain hidden, which means they're recessive. So this was true for the traits he tested, such as P shape, the round shape is dominant, or if one or both alleles specify the round shape, then the P is round. If both of the alleles specify wrinkles, then the P is wrinkled. And then he found similar results in the other 6 traits that he looked at. So here's an example. So this is the cross between 2 homozygotes. So this is the dominant trait, this is the recessive trait. So all of your crosses in the first generation would all have the genotype big R, little r. For the next generation, so this is crossing these heterozygotes, so R, big R, little r, with each other. Then again, if you draw the Punnett square, you'd find a 3-to-1 ratio. And so this is what this table is demonstrating, and these are his results. So he's done very large numbers of these crosses to come up with these estimates. So what type of role why is this important in terms of human traits? So when we talk about a Mendelian phenotype, we're talking about a gene or a loci that encodes for 1 trait. So if something is autosomal dominant, this loci is on the autosome, so, and this is having a dominant role. So an affected person will have a 50% chance of passing on the trait. If it's autosomal recessive, then both parents are unaffected. But if they're both carriers of this recessive allele, then there's a 25% chance that the child will be affected. And this, of course, is more common in related populations. And then if something is X-linked, then it's typically going to affect males. In saying this, given the limited size of human families these days, it's often very difficult to ascertain or determine the mode of inheritance from a single case and an immediate family. So this is why pedigrees are important, and rare conditions are often a guess. But assignment of mode of inheritance is obviously important for risk estimates in genetic counselling. But this is specific for Mendelian phenotypes. This is the online Mendelian inheritance in man database. This is just an example of an online database that has a catalogue of variants which are associated with reported to cause disease. It's very regularly updated. And there's another one called HGMD, which stands for Human Gene Mutation Database. This is a professional curation. And I guess, if you're looking up a particular gene or a particular variant to see if it's associated with a related or a more severe trait that you might be interested in, it's important to note that a lot of these databases haven't recognized how common rare variants are. And so you still need to be critical of the evidence that's in here. And I think the paper that best highlighted this was from Daniel MacArthur and Moncol Lek, who were part of the Exome Aggregation Consortium. And so they were very interested in diagnosing rare Mendelian diseases from exome data. And they found, so they were using exome data because exome data specifically looks at the protein coding regions of the genome. And even though they were using parents and a child, in a lot of cases, they found it was very difficult without a large reference set. And so they actually jointly processed over 60,000 exome sequences from multiple different ancestry populations. And interestingly, they found a very high burden of Mendelian disease causing variants in each individual. So none of these exome cohorts they were collecting were actually enriched for disease, but on average each person had over 50 of these variants. So it demonstrates that there's a lot of inaccurate reports in the literature, and they looked at just those with a minor allele frequency of over 1% and demonstrated that over 95% of these cases had insufficient evidence and were not pathogenic. So literature and database errors are prevalent, and there's lots of false reports of pathogenicity and evidence of incomplete penetrance. So when we talk about incomplete penetrance, all we're saying is that, well, we think this loci is associated with a disease, but in this particular person, it hasn't seemed to demonstrate that. So it's, yeah, so you really need good evidence, which requires careful segregation and case control analyses. I've just got an example here of what I thought might be a typical Mendelian disease, which relates to intellectual disability. Agenesis of the corpus callosum, so this is the corpus callosum here in the brain. These are also associated with other malformations and intellectual disability, and described here are 11 different patients, and they all seem to have different variants, all within the same ARID1B gene, most of which are de novo. So they've got the proband, which is the person affected, the mother and the father. These are the chromatograms to show the sequencing results, and here described is the C means the change in the DNA, and then they've got the protein change. And so what they're showing here is that there are many variants that are all in the same gene that seem to be causing this severe phenotype, which aren't present in the parents, apart from one patient where they also say that there was that the mother was also likely affected, and they also had described evidence of the impact of ARID1B in a mouse model of the disease, which showed similar symptoms. So they described there's many variants, over 70 single nucleotide variants and copy number variations, which explain a proportion of diagnosed cases. And so different variants in the same gene can cause different severity in the phenotype, but there is a commonality across the patients, and there is significant evidence in support of this, I think. And obviously with Mendelian conditions, it can also be modified by genetic background or environmental effects as well.
I think I'm a little confused about, like, so today, how do we define something that's Mendelian? Like, would it just all has to be in the same gene? Like any one mutation that happens on a single gene that causes some sort of effect to some degree is Mendelian?
Yeah, so it's a trait that can be described by a single variant in a gene. So, and yeah.
I guess, so like my question sort of is like, with these you said that sometimes different, it's like actually different like base pairs that are changed.
Yeah, that's right.
And I also correlate with like different severities of the disease, right?
Yep.
So we could maybe imagine that like someone actually who's really unlucky gets 2 of these base pairs changed, right? And they kind of have like a super bad version of this like corpus callosum disease. Is that true?
Yeah, that's possible.
And we would still call that Mendelian though?
Yep.
Okay.
Yeah. It depends. So it can also, your interpretation of that, it depends on what allele it's on as well. So obviously there's recessive. So if it's the same variant that's getting inherited, or if they're in different positions in the genome and then they're on different alleles, then both alleles are kind of damaged. So then that's what you refer to as a compound heterozygote individual. And that you'd expect that to be worse than if you had 2 not so good variants on the same allele. So that also can impact on your interpretation. So the last part, I'm just going to talk about more complex and how do you relate Mendelian laws to complex traits. So in the 1880s, Francis Galton quantified the resemblance between relatives for traits like height, and I think we've heard this from David's talk this morning, but Mendel's laws were rediscovered in the 1900s. However, whether the hereditary or evolutionary properties for a trait like human height were the same as Mendel's P's was actually a source of debate at the time. So there was a real incompatibility between a blending inheritance and also natural selection. So Fisher in 1918 was really able to reconcile Mendelian theory in polygenic traits, and so he was able to derive the theory of resemblance between relatives due to their genetic covariance at single and multiple loci. He actually defined the term variance and showed how phenotypic variance could be partitioned into meaningful genetic and non-genetic sources of variance without knowing anything about the underlying genes. And then he actually concluded that the facts of biometry are consistent with a theory of cumulative Mendelian factors, and that the percentage of variance due to heritable factors, so what we now call heritability, could be estimated numerically from observations of the resemblance between different pairs of relatives. So when we're talking about, this is the usual Mendelian concept, so you've got one gene or one loci influencing one trait. So these are more simple ways to think about a Mendelian trait. Then you've also got pleiotropy, so this is when a single gene can influence two different traits, and you can get genetic correlation between traits, and we know this exists. And then you can also have polygenic traits. So this is things like height, for example, where you can get two genes or many, many more influencing a single trait. Yep.
If I've got a polygenic trait, is that more or less likely to exhibit pleiotropy? So if you've got many different genes affecting one trait,
Yep.
is it more or less likely that those genes will affect? There will be a genetic correlation between that trait and another trait.
I don't. So if we take the example of height, I'm not sure how much overlap there is with, I'm assuming there's a bit of overlap between height and BMI, but I don't know what proportion that is relative to a different complex disorder like type 2 diabetes that also would have overlap with BMI. So I'm not sure how to say them. If more genes are related, then they're more likely to be related with other traits. I'm not sure.
I guess what I'm
I think it might depend what trait,
if I know
how complex it is.
polygenic, like how polygenic a trait is,
Yeah.
can I infer something about how likely pleiotropy is to be a problem when I'm looking at the genes that affect that trait?
I'm not sure. Maybe these guys want to comment.
I mean, I think the answer is maybe, but
it
it
must
depends
be
on the
systematically
details.
though.
Well,
And
but
to some like in extremes.
yeah, but if you think about like a gene that codes for a single thing that's very fundamental, like something like something that matters for every cell in the body or every process, and I don't know what a good example is, but something like that would be highly pleiotropic because it's going to affect all sorts of different things, even though it's a single gene. But if you had a gene that we know, if it's one of these genes that affects eye color, and we think, and that's the primary function of the gene, then I think you could build a plausible case, maybe, if you know the details, which I don't. If you know the details about what the function of that gene is, you might be able to build the case that it's unlikely to have, any effects that it has on other traits are probably orders of magnitude smaller than the effects that it has on eye color. Whereas for highly polygenic traits, I think there's virtually always going to be pleiotropy, because just by the virtue of the fact that there's many genes involved, and genes typically do multiple things, it's
Yeah, I mean, because if you have a trait where 60% of SNPs are associated with the trait, and you have another trait where 60% of SNPs are associated with the trait, necessarily there must be some that are both right, and some there's
Does does pleiotropy like differ from just sort of like regular sort of LD in that like they're part of the same causal pathway, or like I guess what like because there's genes that are just sort of like associated with one another. Like oftentimes when you have one gene, you have another gene, and those genes might have independent effects. Like so yeah, and they're kind of unrelated. So is the difference with that in pleiotropy just that pleiotropy is saying that like the causes are somehow entangled?
I think pleiotropy is one of those words that tends to mean different things in different contexts, and people use it differently. But I think the way that Fleur is using it on the slide, which is a standard way of using it, it's exactly what you said. That it's the same gene doing multiple things, not correlation between a gene and some other gene that do different things.
Linkage disequilibrium literally has to do with the structure of the chromosomes. It has to do with genes physically being close to each other. This is about like what the gene does and genes can regulate. So they're really independent concepts.
Yeah, but you could worry that like if you had two SNPs that are near each other and so they're highly correlated, but one SNP's the actual causal SNP for a certain trait and the other SNP's the causal SNP for some other trait, and you did like a GWAS, then both of those SNPs are going to be associated with both of the traits, even though there's not kind of pleiotropy in this sense here, because the actual causal, there's not overlap in the causal SNP. And so and I think that's kind of what Dan was saying. It's like, well, in different settings, sometimes when people say pleiotropy, they mean this kind of really broad common association thing, or sometimes we mean this very specific thing here.
Seems like that's a theme with, and maybe it makes sense, right, just because it's kind of different fields sort of intersecting with one another, but like with how people sort of construct the environment versus like genetic effects, or like with how people think of pleiotropy, but just sort of different contexts, the same word sort of meaning slightly different things.
Yeah, it's a pain.
Okay.
Well, I mean, for the statistical issues, pleiotropy and linkage between things that are really close together could create similar statistical issues.
Yeah, yeah. And often, since you don't know what the causal variant are, you can't distinguish anyway between them. And so, for convenience, you might just call that pleiotropy.
Yep.
Yeah, in two recent comments I heard linkage and linkage disequilibrium used interchangeably. This is not proper usage, I guess. I hate to get pedant, but I mean, linkage just means that the two SNPs are sufficiently, well, the two parts of the genome, points of the genome are sufficiently close together that they tend to be co-inherited. So, for example, if you look within a single family, and the recombination probability between these 2 points of the genome is not the asymptotic 0.5, then we say that they're linked. Linkage disequilibrium refers to a correlation in the population between what you see at the 2 points. And typically, LD extends much shorter than linkage. So within the same family, like it could be that like, you know, a SNP at one end of the chromosome and a SNP in the middle of the chromosome are actually linked because they'll tend to travel together. However, it will almost certainly be the case that in the population, a SNP in the middle of the chromosome and a SNP at the end are going to not be linked.
Cool.
It seems like linkage disequilibrium would have something to do with how long ago a mutation happened. So, and not this distance on the chromosome. I mean, is it? I mean, when you say disequilibrium, it makes it sound like if a variant has been around for a long enough time, that you ought to be in equilibrium. But is that wrong?
I think you're basically right. So linkage disequilibrium was a term introduced by Richard Lewontin in 1960, and the basic idea is that so there's a correlation now, but as long as recombination occurs, with the passage of time, it will disappear, and at that point, I guess that's the equilibrium.
I think Raymond's going to give, I was going to say, I think Raymond's going to give a lecture on LD, is that right?
Yeah,
So-
I mean, LD score, I think, which we'll probably talk about LD, I suppose. But
Okay.
The point is that I think that any correlation that exists at the population level, the linkage disequilibrium, is a disequilibrium phenomenon in the sense that it would all disappear with enough time.
In the limit, okay.
But the distance between each other probably determines how quickly it approaches the.
Yeah.
Like if they're right next to each other, it's going to take a long time for that to go away.
All right, just 2 more slides, I think.
Is it kind of
Yeah.
decay in the distance?
Yeah, because it has to do with this crossing over. The crossing over is what eliminates the correlation over time, and the crossing over, you know, the crossing over is gonna
Reduce the co-segregation, essentially. Reduce the chance of co-segregation, so just.
yeah.
Yeah.
Sorry. But if there was a subpopulation that's isolated, would the convergence still happen ultimately?
Well, it depends if in everybody, if every person in that isolated population has the same sequence in a particular place, it's going to take, it's essentially not going to ever go away, that correlation there is, except for mutations, which are extraordinarily rare events. So that it'll take an extremely long time for the correlation to go away. So if you assume away mutations, then you'll never, you know, you'll always have that linkage to equilibrium in that isolated population.
I mean, they'll go away within the population over time.
Yeah.
Right. Just the,
Okay. So there are a few exceptions to Mendelian disease, or Mendelian theory, basically. We can have incomplete penetrance, as I've talked about. You can also have variable expressivity, and this can relate to things like expanded repeats. So incomplete penetrance is just talking about the probability that a person has the variant will express the phenotype, so this isn't always a given. And it can also vary based on age of onset. For scenarios such as expanded repeats, in this diagram here, this is an example of what we call term anticipation. So basically, with each generation you get an earlier age of onset and a more severe phenotype, and this is because basically the expanded repeat isn't, there's a issue with replication, so it kind of tends to stutter, and then over generations that expansion repeat can get larger over time. And so this is common in genetic disorders such as Fragile X, Huntington's and myotonic dystrophy. And of course there's also the effects of other genes and the environment. Genomic imprinting is known to occur. An example being chromosome 15 deletion. So certain characteristics are dominant, but they manifest only when inherited from the parent of the same sex. And so genes are somehow imprinted with their parental origin. And so examples of this is things like Angelman syndrome and Prader-Willi. So I'll let you look that up in your own time, but obviously these are kind of exceptions to these laws. So they're not common, but they do exist.
is there any, like, can we look at the gene and and know that it came from the, like, is there a structural thing that we know about that this imprinting represents, or like?
How can you determine? I don't know the best way to pick it up.
That it has to do with DNA methylation, and that if you chemically remove that, imprinting vanishes, but it's not entirely clear.
Yeah. You can have changes in chromosomes, so obviously Down syndrome would be the most well-known example. So you've got an extra chromosome 21. And then you've got de novo situations where it's not present in the parent's somatic cells, but it could have occurred in the parent germ cells or during cell division. So this is the final slide, just a summary. We've talked about mitosis and meiosis, talked about Mendel's laws. Independent assortment and segregation are established principles that govern hereditary. Mendelian disease refers to a genetic architecture of a single trait explained by one gene or one locus. Protein coding areas of the genome are enriched for these loci. And this is actually independent of, I guess, probably a lot of the sequencing studies have kind of focused on this protein coding region, but I think in studies where even when you control for this ascertainment bias, that these Mendelian loci are still enriched in this protein coding region relative to other areas of the genome. And Fisher in 1919 produced seminal works on the theory of genetics, and of course is worth reading when you get the chance. And of course there are exceptions to Mendelian properties. So that's it for today. Do more tomorrow.